All right, guys. So this video is going to be relatively short. It's just about what viruses are and kind of the specifics of what you should be able to understand from these. And for the most part, there's not a whole lot that we have to fixate on besides a couple of key points. So um, a virus is going to have its own genome. That basically means that we have different types of viruses, different types of species. Um, They do evolution just like any other thing with DNA. But the difference about a virus is that they can only reproduce within a host cell. So the basic structure of them is there's going to be some, in terms of genetic material here, either DNA or RNA. And these things in purple on the exterior are going to be proteins. So um, in terms of this little video, I just want to kind of recap where viruses fit in terms of their evolutionary existence. And this is a nice little comparison of everything that you probably should be able to tell me whether or not you have a eukaryotic cell, a prokaryotic cell, and a virus. So the big things here that I at least want to mention is when you look at viruses, there's no nucleus or nucleoid, but there is some DNA or RNA. So don't think that viruses don't have genetic info. They most definitely do. Um, but basically, they're just these two things, DNA and RNA and a protein code. They don't have ribosomes. They're going to use whatever cell they infect with. They're going to use their ribosomes. They are going to use the ATP that the host cell has, and they're incredibly simple. Now, just kind of revisit prokaryotes versus eukaryotes. Remember, eukaryotes have that linear DNA chromosomes. Prokaryotes, things like bacteria, they have that circular DNA. So I just want to kind of remind you guys of the main three things about these viruses. Basically, they have a couple things. The nucleic acids, either DNA or RNA. Some viruses are going to be double-stranded, some are single-stranded, some are double-stranded RNA, which is really weird. And a lot of them, what are called retroviruses, are going to be single-stranded RNA. So all kinds of different genetic information that these viruses can actually have and possess. So basically, in terms of this, the main thing that all these viruses are going to share is they're going to have what's considered a capsid, which is a protein coat. And basically, it's just this structure that allows it to actually infect the host cell. So it surrounds the capsid of those viruses. There's going to be these little glycoproteins, which again is just carbohydrates and proteins used for cell-to-cell recognition. In some of these viruses, like a bacteriophage that infect bacteria cells, they're going to have a tail. So this is something that I think is worth mentioning here. Each one of these are really broad classifications of viruses. So this one, if you look at the difference, the main one that we talk about a lot in this class is going to be bacteriophage. These terrifying looking things. There's some DNA inside of there. There's a protein coat. And then here's those tails that I just mentioned. Now, these are the tails that can actually infect a cell. But if you look at other things like an influenza, I want you to pay close attention to these glycoproteins on the surface because you never get the flu more than once, or I should say the same type of flu, but you obviously do. But the reason being is because these glycoproteins evolve, and basically it allows the cell to infect your cells again. So basically what you want to at least understand here is that it's these receptor or these surface proteins, right? The glycoproteins that allow the cell to infect a specific type of a cell, right? So if you're looking at this one, this influenza virus, these receptor proteins can actually infect your eukaryotic cells, whereas the bacteriophages, these little tails, allow it to only recognize these prokaryotic cells, and that's how they infect those. So just showing you different classes of viruses and their protein codes and how they can reproduce. The one that you do have to know a little specifics about are these bacteriophages. Now, they're often going to be referred to as phages for short, but all of these bacteriophages, these terrifying looking things, those are going to be the ones that can only infect bacteria. So this is showing you a shot of these viruses infecting a bacteria or E. coli cell. So they have pretty intense capsids in terms of the protein coat. But like I said, the main things that you want to know is that inside there's going to be some genetic material. And these bacteriophages can only infect bacteria, hence the name bacteria. So it's really simple if you remember it like that. Now, let's talk about kind of how these things reproduce. This just kind of shows you that you have these viruses inside. This is showing a eukaryotic cell, and I know that because I see a nucleus, I see a couple of mitochondria. But basically, it does endocytosis. So this is not one of those bacteriophages. It's just showing you that there's these little receptors that these viruses can detect. So there's going to be these docking proteins, right? These glycoproteins, like I mentioned earlier, are going to allow these things to bond to the cell and enter via endocytosis. So, and then when you watch what happens here, then all of a sudden this virus is exposed and it's going to take over the cell using the machinery. It's going to use the ribosomes. It's going to use all the ATP to make copies of itself. So that's how it's really like a lock and key fit, right, between the host cell and the receptors. So let's talk a little bit more about the bacteriophages and what you need to know. This is going to show you bacteria. So this is some type of prokaryotic cell. But watch what happens here and how this works is that these bacteriophages are going to dock. They're going to inject their genetic information, whether this is DNA or RNA. And then what it does is it's going to make a bunch of different copies of itself. Not only replicating the DNA, it's making more proteins. And it makes so many of them that the bacteria cell often pops. So it kills the host cell after a while. So let's just look at this really quickly via mastering biology. So this one is a lot shorter and a lot clearer animation. So just reminding you that this is the prokaryotic genome, right? This is the circular DNA. There's the phage. This one doesn't show the tails, but it does have tails. So it injects its DNA, and now that DNA is going to become part of this DNA of the bacteria's prokaryotic cell, right? So remember, this is called transduction in terms of how bacteria genomes can change. So basically what this means now is all of a sudden when the RNA polymerase is going to read this, it basically has no proofreading. It just is going to read that genome and it's making copies of the virus itself. So now as you're doing this, every time that host bacterium reproduces, it's going to replicate that phage DNA again and again and again and again, okay? Now, so far, it doesn't look like there's been any harm done because as this bacteria is replicating, it's just replicating that new genome. However, and this is what's called the lysogenic cycle, and then we'll talk about the lytic cycle here in just a second. But occasionally the phage DNA in the bacteria chromosome initiates the lytic cycle. So watch what happens with the lytic cycle. This is when it actually starts to express or read all of that DNA from the virus. And if you see what's happening here, as it's making more and more and more of those copies, it makes a bunch more and the cell actually pops, and then it's going to spread that DNA to another host cell. Okay, so anytime a cell essentially pops, we're going to refer to that as being lysed, L-Y-S-E. So basically what you saw here is that all of a sudden when it was making copies of itself like this, when it's just making copies of the viral genome, okay, this is going to be the lysogenic cycle. So it's just basically spreading that viral DNA as the bacteria replicates. But as soon as it starts to express it, it's going to enter what's called the lytic cycle, where it's going to make more copies of the proteins and more genetic information with the virus, and then the host cell essentially is going to lyse. So it makes more copies, the host cell is going to burst, then the virus is going to go and infect another bacteria. So kind of interesting component here, and this just shows you, like I said, the lysogenic cycle can get called over to the lytic cycle. And so that is the one specifics about these bacteriophages you did have to know in terms of the lytic versus the lysogenic cycle. So understanding that, hopefully it's not too terrible, the lytic versus that lysogenic cycle. And the last thing we do have to mention here is what are considered retroviruses. And retroviruses are things like HIV. And the interesting thing about these retroviruses is they use an enzyme called reverse transcriptase. And so let's explain why retroviruses are somewhat tricky to catch and treat. So if you look here, these retroviruses, you're seeing a similar theme, right? So you have your protein coat, right, all along the exterior. But the interesting thing about this is that it actually has RNA. So when we have what's called a retrovirus, it actually doesn't do central dogma. It actually goes from RNA back to DNA. Very few things actually do that. And this is showing you within the virus itself, it has that enzyme reverse transcriptase, and that allows it to actually go from RNA back to DNA. So that's something that is a little interesting about these retroviruses. So in terms of that, let's kind of take a look and see how this would infect your cells. And this just kind of shows you, right? You have the protein coat. As soon as it enters into the cell, it's going to have that viral DNA. It uses that enzyme reverse transcriptase, and then all of a sudden, that RNA DNA hybrid becomes DNA using that enzyme. So it actually, I don't want to say violate, but it goes from RNA to DNA. Then that DNA actually gets inserted into a chromosome. And basically what that does is that now all of a sudden it can make more copies of itself, makes more copies of the mRNA, makes more copies of the protein, and then it takes over the host cell. Now, if this is HIV, there's other issues that we aren't going to talk about now, but things like how it impacts your immune system and stops it. This is just talking about the main process of what's a retrovirus. What you need to know is that it actually goes from RNA back to DNA, then that DNA becomes part of the chromosome. So in terms of evolution of viruses, viruses really, they don't fit a definition of living things because they can't reproduce within their cells, but they definitely evolve because that genetic information changes over time. So that's why the flu keeps popping up year after year. Why you get a flu shot is because it evolves in the sense of random mutation. So understanding that, just to kind of wrap this up, let's see if this gives us any viable information about retroviruses. I think it's just testing their RNA molecule. Well, let's take a look at this one. So this one's showing you, yeah, it's injecting the virus is doing endocytosis. It's using that enzyme reverse transcriptase. It's making a copy, going from RNA back to DNA, and then that DNA is going to actually go ahead and infiltrate the nucleus and become part of the double-stranded DNA in the chromosome. So, and that's what we refer to as a provirus, not something you need to know specific, but it just means that when we say it's a provirus, it's just viral DNA that inserts into a host gene. So, and that's where it gets sometimes difficult to detect and treat when you look at the actual genetic information. So this just shows you once that DNA is actually embedded in the nucleus, you're making more copies of the RNA. That RNA is going to be transcribed and right, the DNA is transcribed, and then it's going to get translated here to make more proteins. You make the code, and then you make the RNA copy, and then this is going to go and infect another cell. So that's just kind of a basic, simple overview of what these viruses are, and we will see you guys in the next video.